Welcome to our deep dive into metabolic pathways, cornerstone concept in biomedical science. Metabolism refers to the sum of all chemical reactions that occur within living organisms to maintain life. These reactions are organized into complex networks known as metabolic pathways. Understanding these pathways is crucial for grasping how our bodies convert food into energy, build essential molecules, and eliminate waste products. In this video, we'll explore the major metabolic routes, their significance, and how they interconnect to sustain life. Let's begin by laying the foundation for what metabolic pathways are and why they matter in the context of health and disease. Metabolic pathways are sequences of chemical reactions, each catalyzed by a specific enzyme that transform molecules within a cell. These pathways can be linear, circular, or branched, and they often share intermediates with other pathways. The organization of these reactions allows cells to efficiently regulate energy production, biosynthesis, and waste removal. By understanding the structure and function of metabolic pathways, scientists can better comprehend how cells respond to different physiological conditions and how disruptions can lead to disease. Metabolism is broadly divided into two categories: catabolism and anabolism. Catabolic pathways break down complex molecules into simpler ones, releasing energy in the process. Anabolic pathways, on the other hand, use energy to build complex molecules from simpler ones. This balance between breaking down and building up is essential for maintaining cellular function and overall health. Both types of pathways are interconnected, ensuring that the body can adapt to varying energy demands and nutrient availability. Adenosine triphosphate, or ATP, is often called the energy currency of the cell. It stores and transports chemical energy within cells, fueling countless biological processes. Most metabolic pathways are designed to either produce or consume ATP, making it central to metabolism. When ATP is broken down into ATP and inorganic phosphate, energy is released, powering everything from muscle contraction to nerve impulses. The continuous regeneration of ATP is a primary goal of many metabolic pathways. Glycolysis is one of the most fundamental metabolic pathways. It takes place in the cytoplasm of cells and involves the breakdown of glucose into pyruvate, generating a small amount of ATP and NADH in the process. Glycolysis is unique because it does not require oxygen, making it essential for energy production in both aerobic and anaerobic conditions. The pathway consists of 10 enzyme-catalyzed steps, each carefully regulated to meet the cell's energy need. After glycolysis, pyruvate enters the mitochondria and is converted into acetyl-CoA, which then feeds into the citric acid cycle, also known as the Krebs cycle. This pathway is a series of chemical reactions that generate high-energy electron carriers, such as NADH and FADH2, as well as a small amount of ATP. The citric acid cycle is central to aerobic metabolism and provides the building blocks for many biosynthetic processes. Electron transport chain, ETC, is the final stage of cellular respiration and takes place in the inner mitochondrial membrane. Here, electrons from NADH and FADH2 are transferred through a series of protein complexes, ultimately leading to the production of a large amount of ATP. Oxygen acts as the final electron acceptor, combining with electrons and protons to form water. The ETC is highly efficient and is responsible for the majority of ATP generated in aerobic organisms. Oxygen is scarce, cells can switch to anaerobic metabolism to produce energy. In this process, pyruvate from glycolysis is converted into lactate in animals or ethanol in yeast, allowing glycolysis to continue. Although anaerobic metabolism generates much less ATP than aerobic pathways, it is crucial for survival during intense exercise or low-oxygen environments. This flexibility highlights the adaptability of metabolic pathways. Gluconeogenesis is the process by which cells synthesize glucose from non-carbohydrate sources, such as amino acids and glycerol. This pathway is especially important during fasting or intense exercise when glucose levels are low. Gluconeogenesis primarily occurs in the liver and helps maintain blood sugar levels, ensuring a steady supply of energy for the brain and other vital organs. Glycogen is the storage form of glucose in animals, primarily found in the liver and muscles. Glycogen metabolism involves two main processes: glycogenesis, the synthesis of glycogen from glucose, and glycogenolysis, the breakdown of glycogen to release glucose. These pathways are tightly regulated to balance energy storage and release, especially during periods of fasting or physical activity. Fatty acid oxidation, also known as beta oxidation, is the process by which fatty acids are broken down in the mitochondria to produce acetyl-CoA, NADH, and FADH2. This pathway is a major source of energy, especially during prolonged exercise or fasting. The products of fatty acid oxidation feed into the citric acid cycle and electron transport chain, linking lipid metabolism to overall energy production. Contrast to fatty acid oxidation, fatty acid synthesis is an anabolic pathway that builds fatty acids from acetyl-CoA and malonyl-CoA. This process occurs mainly in the liver and adipose tissue and is essential for storing excess energy as fat. The pathway is regulated by nutritional and hormonal signals, ensuring that fatty acid synthesis is active when energy and building blocks are abundant. Amino acids, the building blocks of proteins, can also be used for energy production or converted into other biomolecules. Amino acid metabolism involves deamination, where the amino group is removed and the carbon skeleton is funneled into various metabolic pathways. This process is vital during periods of fasting or intense exercise, when proteins are broken down to meet energy demands. Urea cycle is a crucial pathway for removing excess nitrogen from the body. When amino acids are broken down, their amino groups are converted into ammonia, which is toxic. The urea cycle, primarily in the liver, converts ammonia into urea, a less toxic compound that can be safely excreted in urine. This process is essential for maintaining nitrogen balance and preventing toxicity. Pentose phosphate pathway is an alternative route for glucose metabolism. It generates NADPH, a reducing agent used in biosynthetic reactions, and ribose 5-phosphate, a precursor for nucleotide synthesis. This pathway is especially active in tissues involved in rapid growth or detoxification, such as the liver and red blood cells. Metabolic pathways do not operate in isolation. Instead, they are highly integrated, with intermediates and products of one pathway feeding into others. This integration allows cells to efficiently allocate resources and respond to changing energy demands. For example, during fasting, the body shifts from using glucose to fatty acids for energy, demonstrating the flexibility and interconnectedness of metabolism. Hormones play a central role in regulating metabolic pathways. Insulin, glucagon, adrenaline, and cortisol are just a few examples of hormones that influence metabolism. Insulin promotes glucose uptake and storage, while glucagon stimulates glucose release. These hormonal signals ensure that energy production and storage are matched to the body's needs, maintaining homeostasis. Enzymes are the catalysts that drive metabolic reactions, and their activity is tightly regulated. This regulation can occur through changes in enzyme concentration, allosteric modulation, or covalent modification. By controlling enzyme activity, cells can rapidly adjust metabolic flux in response to internal and external cues, ensuring efficient and balanced metabolism. Feedback inhibition is a common mechanism for regulating metabolic pathways. In this process, the end product of a pathway inhibits an earlier enzyme, preventing overproduction. This self-regulating system helps maintain metabolic balance and conserves resources, ensuring that cells only produce what they need. Cells compartmentalize metabolic pathways within specific organelles such as mitochondria, cytoplasm, or peroxisomes. This spatial organization allows for specialized environments, efficient substrate channeling, and separation of incompatible reactions. Compartmentalization is a key feature of eukaryotic cells and enhances metabolic efficiency. Metabolic flux refers to the rate at which substrates and products move through a metabolic pathway. It is influenced by enzyme activity, substrate availability, and regulatory signals. Measuring metabolic flux helps scientists understand how pathways are controlled and how they respond to physiological changes. Genes encode the enzymes that catalyze metabolic reactions. Mutations or changes in gene expression can alter enzyme levels and activity, impacting metabolic pathways. Genetic control is fundamental to understanding inherited metabolic disorders and how genetic variation influences metabolism. Disruptions in metabolic pathways can lead to a variety of diseases known as metabolic disorders. These include diabetes, phenylketonuria, and mitochondrial diseases. Understanding the underlying metabolic defects is essential for developing targeted therapies and improving patient outcomes. Cancer cells often reprogram their metabolism to support rapid growth and survival. They may rely more on glycolysis, even in the presence of oxygen, a phenomenon known as the Warburg effect. Studying cancer metabolism provides insights into tumor biology and potential therapeutic targets. Obesity is closely linked to metabolic pathways involved in energy storage and expenditure. Excess calorie intake leads to increased fat storage, while metabolic adaptations can make weight loss challenging. Understanding the metabolic basis of obesity is key to developing effective interventions. Diabetes mellitus is a metabolic disease characterized by impaired glucose metabolism. In type 1 diabetes, the body cannot produce insulin, while in type 2, cells become resistant to insulin's effects. Both forms lead to elevated blood sugar levels and can cause serious complications if not managed properly. Inborn errors of metabolism are genetic disorders caused by defects in specific enzymes. These defects can lead to the accumulation of toxic substances or the inability to synthesize essential molecules. Early diagnosis and dietary management are often critical for preventing severe outcomes. Metabolomics is the comprehensive study of metabolites within a biological system. By analyzing the small molecules produced during metabolism, scientists can gain insights into cellular function, disease states, and responses to treatment. Metabolomics is a powerful tool in biomedical research and personalized medicine. Cells constantly monitor nutrient availability through specialized sensors and signaling pathways. These systems help adjust metabolic activity to match nutrient supply, ensuring optimal growth and survival. Disruptions in nutrient sensing can contribute to metabolic diseases and aging. Circadian rhythms are biological clocks that regulate metabolic pathways according to the time of day. These rhythms influence hormone release, enzyme activity, and nutrient metabolism, helping organisms anticipate daily changes in their environment. Disruption of circadian rhythms can negatively impact metabolism and health. Physical activity has a profound effect on metabolic pathways. Exercise increases energy demand, stimulates glucose uptake, and promotes fatty acid oxidation. Regular activity improves metabolic health, enhances insulin sensitivity, and reduces the risk of metabolic diseases. As we age, metabolic pathways undergo changes that can affect energy production, nutrient utilization, and cellular repair. These alterations contribute to age-related diseases and reduced physiological function. Research into aging and metabolism aims to identify interventions that promote healthy aging. During prolonged fasting or starvation, the body adapts by shifting metabolism to conserve energy and maintain vital functions. Glycogen stores are depleted, fatty acids become the primary fuel, and ketone bodies are produced for the brain. These adaptations highlight the flexibility of metabolic pathways in response to extreme conditions. Thermogenesis is the process of heat production in organisms, often linked to metabolic activity. Brown adipose tissue, for example, generates heat by uncoupling ATP production from the electron transport chain. Thermogenesis plays a role in temperature regulation and energy balance. Metabolic pathways that involve oxygen can generate reactive oxygen species, ROS, which can damage cells if not controlled. Antioxidant systems help neutralize ROS and protect against oxidative stress. Imbalances in these systems are linked to aging and various diseases. Body uses specialized metabolic pathways to detoxify harmful substances such as drugs and environmental toxins. The liver is the primary organ for detoxification, using enzymes to modify and eliminate toxins. Efficient detoxification is essential for health and protection against chemical damage. The gut microbiome, composed of trillions of microorganisms, plays a significant role in host metabolism. Microbes help digest food, produce vitamins, and influence metabolic pathways. Disruptions in the microbiome can impact energy balance and contribute to metabolic diseases. Plants have unique metabolic pathways such as photosynthesis, which converts light energy into chemical energy. They also synthesize a wide range of secondary metabolites for defense and communication. Studying plant metabolism provides insights into energy flow and adaptation in nature. Metabolic engineering involves modifying metabolic pathways to improve the production of desired compounds, such as biofuels, pharmaceuticals, or food ingredients. By manipulating genes and enzymes, scientists can optimize pathways for industrial and medical applications. Systems biology integrates data from genomics, proteomics, and metabolomics to model and understand metabolic networks as a whole. This holistic approach reveals how pathways interact and respond to changes, advancing our understanding of health and disease. Many drugs are metabolized by the body's enzymes, affecting their efficacy and toxicity. Understanding metabolic pathways is crucial for drug development, dosing, and predicting side effects. Personalized medicine often considers individual metabolic differences to optimize therapy. Nutritional metabolism examines how the body processes nutrients from food, including carbohydrates, fats, and proteins. Proper nutrient metabolism is essential for growth, repair, and energy. Imbalances can lead to deficiencies or excesses, impacting health. Environmental factors such as temperature, diet, and toxins can influence metabolic pathways. Adaptation to environmental changes is essential for survival and health. Studying these influences helps us understand how organisms cope with stress and maintain metabolic balance. Research in metabolic pathways continues to advance, with new technologies enabling deeper insights into cellular processes. Emerging fields like synthetic biology and precision medicine are poised to revolutionize our understanding and manipulation of metabolism for health and industry. Summary: metabolic pathways are intricate networks that sustain life by managing energy flow, biosynthesis, and waste removal. Their regulation is vital for health, and disruptions can lead to disease. By studying these pathways, biomedical science continues to unlock new possibilities for diagnosis, treatment, and prevention. Thank you for joining this exploration of metabolic pathways and their significance in biomedical science.